try to explain as simply as possible what we have done over 25 years with Stargardt genetics treatment and where we are now. If some of my presentation is too complicated or there are issues with seeing it, so again, as Bhavna said, it will be recorded, and you can always ask questions about it. So is the screen now up normally, Bhavna?
It's on 2.
It's still on 2.
Yeah,
yeah?
2. We need to.
Okay, let me try again. How about now?
Yeah, and then some of the bit of the writing's missing. So
Writing is missing.
like we did last time, I think, when you just showed it. So I think you need to make the screen a bit smaller, maybe, what you did last time.
No, last time I did not do much differently. So there is a problem with?
We can only see some of the writing.
Last time it was the third time you tried, it shared properly.
Okay, let me try again. So now somehow it is stuck here. This is always the case when that it works fine when you rehearse, and then something is different when you...
Oh, there we are. I can see it now.
So can you see it properly or not?
We can see, but we can see the writing on the left-hand side as well. So the actual slides. So you need to go onto full screen.
Yeah, it's not in presentation mode yet.
Not in presentation. Yeah, now it is. It's perfect. But
Excellent.
Yeah.
Now it's fine?
Yes.
Yes, there's a little window that says, please move this window away from the shared application.
Yeah, I don't know what that is. No.
That might disappear if you go on to the next slide.
No.
Oh, no, it's not there. If you can.
Okay.
Oh, it's gone. It's gone. Brilliant. We're ready.
I don't think it's gone, but...
Oh, no, it's not gone.
I'm just trying to see how this one... I've never seen this window before, you know, in all my Zoom. No, it is not working.
Oh, it's gone now. Oh, no, it's gone.
No, it is just in the background, and I don't know why. And actually, I don't know how to get that moved.
He's done it for me. It's
Okay,
where
let
the
me stop the share.
No, I have. Mine is lack of not moving.
What?
Mine's
No, that's right. I should get you fixed.
Ashrafia? She's there. She's on mute now. Okay. Okay, I think third time lucky. Yeah, we're ready to roll.
Okay, let's go. So, yeah, my name is Rando Allikmets. I'm currently at Columbia University in New York, where I've been about 22 years now. And I will tell you the story, some of the history, and some of the most current information about the Stargardt disease, what we know about its genetics, what we know about possible treatment options, and what are we doing as scientists to move this field ahead so the treatment can be found, approved, and administered quickly, and how can we help with that. So a little bit of a background. people are asking where I'm from and where is the accent coming from. So I'm originally from Estonia. This is the map I cut out from Washington Post when I arrived in the US in '91, because nobody knew and still doesn't know much what Estonia is and where it's at. So this is where I was born and spent first 17 years of my life, but the rest of the time I've been traveling all around the world. And as I said, now I am at Columbia, but when I came to the US, I'm actually a cancer researcher by trade, cancer geneticist, human genome researcher. So I was not planning at all to work on any eye diseases by definition. So, but when I arrived at National Cancer Institute, I was tasked to study the ABC transporter gene family in humans. So this is the gene family or protein family that uses the ATP energy to move different substrates through membranes. So this is a family of pumps that really work to move either important or unimportant, useful or bad things throughout membranes, and therefore helping the cells to perform their functions properly. And I will come specifically to that. So since some of these genes are involved in cancer chemotherapy, that's why I was working on this subfamily, and I cloned a whole bunch of them. But at that time, what we were doing, we were at that time cloning a gene. That was mid-90s, was still an art. So nowadays, you know, you do a click on a computer and you have the gene. But then it was a lot of hard manual labor, and we first were pulling pieces of the gene out of the libraries. Then we were mapping them on the genome, on which chromosome it goes. And then we were also trying to do like a standard studies to see where it is expressed, in what organs. And this was done on so-called northern blots, where we had material from all major human organs, you know, heart, kidney, brain, so on and so forth. Now, when I cloned a piece of this gene, we were stymied because we couldn't see it expressed anywhere. And also we had some issues with mapping. But then, so there was a question how we figured out it's a Stargardt disease gene. It was really quite straightforward once we mapped it to human chromosome 1. And the year before that, in '95, the Stargardt region was mapped there. So region mapping is done by linkage analysis in families. And what that analysis does, it looks what region in the genome is shared by all patients, you know, all affected members. So they said, well, Stargardt gene has to be in this piece, but that piece was really big. But now when we mapped the ABCA4 gene, so that mapped smack right in the middle of that region. So it was clear to me that, you know, that could be something. And then I said, aha, that's why we don't see the expression. So it's not expressed anywhere. So we got, and we were in Cancer Institute, we didn't have any materials from eyes. So then we got the human eye RNA, and it was expressed big time. So it is expressed in photoreceptors, it is expressed in RPE, and it's not expressed in any other tissue, pretty much. So it's a very specifically expressed gene and protein that performs a very specific function in the visual cycle. I'll show you a couple of slides about that. So then we called it ABCR. Everybody was joking. I called it after myself as Rando. No, it was retina, was R. But since we were doing, as I said, cloning all human ABC transporters, now there are 48 of them total in human genome. We had to rename the entire superfamily. And so then we made it into subgroups of A, B, C, and E, and then ABCAR became ABCA4 because it is in A, it's a fourth gene in A subfamily. So that is kind of the history. And then, as I said, we figured out it's a Stargardt gene immediately, and then also that it was the first gene we described as involved in age-related macular degeneration. So it got a lot of press. So this is a picture at the time we did clone it. So from Dean's lab, this is Mike Dean, who is was the head of the lab. He looks like a student. Next to him, the guy who looks like a head is actually a student. So, but that picture is about from '96. I mean, we had done many very great discoveries these years. So that was back then at National Cancer Institute. So now to the Stargardt disease. So again, what we know, and we knew it then, it is a recessive disease. Yeah, so we all have 2 copies. All humans have 2 copies of any gene we have. And the recessive disease means that both copies of the gene are mutated. So you get 1 copy. I mean, for any gene, you get 1 copy from your mother, the other from your father. And if you are not lucky, and this is what I call genetic lottery, that, you know, the mother carries 1 of the copies, the other is fine, the father the same. But if by chance, and the chance is 25%, if they get together, the kid is affected with the recessive disease, in this case, Stargardt disease. So Stargardt disease, I'll show you, and this has been the main focus of our research. It's very variable. So it is clinically variable, genetically variable. But it has some features that are quite specific. So you lose your central vision. So usually there is an atrophy, is what we call macular, or even the more central part of the macular fovea. This is where we have most of our cone photoreceptors. So this is where we have central vision, color vision, and so on. And so that part is affected in Stargardt disease. Periphery is most often not affected at all. So that's why it is kind of the direct opposite to another well-known eye disease, retinitis pigmentosa. Yeah, when you start losing the vision from periphery, and then you get, you know, to a tunnel vision. In this case, you lose the central vision, but your peripheral vision mostly stays intact. And again, as I mentioned, it is caused by mutations in ABCA4, which is the photoreceptor specific protein, and it transports vitamin A in the visual cycle. This may be a little too complicated drawing, but it just shows schematically where it is located. It is located on the tips of our photoreceptors that are invaginated into the retinal pigment epithelial cells, and there is this cycle of visual cycle going on. That's what makes us see anything. And ABCA4 protein is important to move a vitamin A derivative that is the main component of this cycle in and out of the cycle. So it is kind of a janitorial task. It cleans up the cycle. And if it is not working, so we'll see this happening. We have accumulation of this toxic material, what is in general called lipofuscin, or sometimes you hear the word A2E, what is a part of lipofuscin. So what you can see in a specific imaging technology called quantitative autofluorescence, that you have these huge accumulations of fluorescent material in your retina. Specifically, it accumulates in the retinal pigment epithelium. And we can use now this method. It was developed at Columbia independently. It was developed also in some other places. But what this does is that you can really quantify the disease process. Yeah, you can see, you know, how bad it is, what it is, and you can also track the progress of disease by that. Now, this said, and I'll show you later, this is not an absolutely universal feature of Stargardt disease. People usually say it is, but you can see here also, I mean, there is a graph on the left side And we all accumulate lipofuscin with age. You can see there is an age curve that goes up, so that's normal. And then the black dots are Stargardt patients, so you can see that many of them, or we can say most of them, have highly elevated of this lipofuscin levels. And what this does is that this is a toxic component for the cells. It really simplistically at some point kills the cells. And this is one way Stargardt disease develops, and this is one way of seeing it. Now, again, a quick cartoon here. What are recessive and dominant disorders? I mentioned that, you know, and this has to do with a treatment I've been talking to you about. So in any normal genome, you have 2 copies of the gene. They are happy, they are working. In recessive disease, they are both not working. Yeah, so then that's why you get the disease. Now this you can treat very nicely with gene therapy, and this is what I would suggest to be the best treatment for Stargardt's. You just add a functional gene. You don't have to do anything too you know, crazy or too complicated. Usually the gene is delivered in viral vectors, like viral particles. There are several of them. I may touch a little bit upon it. But so this is a very well worked out technology. You just take the normal gene, put it into the virus, deliver it to the cell, in this case photoreceptor, and it starts working. And you don't need to replace anything. You just add it, and you don't need to add a lot. You know, sometimes adding, you know, some also has a good effect. So that is the gene therapy. Now, in dominant diseases, I will not go there today. You have this issue where one copy is bad, and that bad copy does everything bad, even to the good copy. So in order to gene adding doesn't work in that case, you know, because the bad copy is still there. What you have to do is you have to really kill that bad one, and so the half of the good one is still quite enough to do that. So these are kind of simplistically the ways you treat any disease, and including Stargardt. But Stargardt, since it's a recessive disease, will focus mostly on that part of treatments. Okay, so now straight to the treatments and things, what we do have and what should we do to get very efficient clinical trials and treatment options. There are many treatment options for Stargardt's in preclinical studies. Some of them have been in clinical trials, and so I'll touch upon them a little bit of this and that. Now the problems are, yeah, so there are many treatment options. Some of them are gene therapy that target directly the faulty gene. Some of them are therapies that target, let's say, that lipofuscin accumulation, what I was talking about. Then there are some other things. There are therapies that are targeting or general well-being of photoreceptors. Yeah, so it really doesn't matter that much what mutation in what gene do you have, but there are some growth factors, some other components that are used to better the health of photoreceptors. So again, this is not a cure, but this is something that, you know, has an effect and prolongs the life of photoreceptors in order for getting in, let's say, with gene therapy once it is available. So again, one issue with Stargardt's and similar diseases is that they are slow progressive diseases in most cases, but clinical trials, they have to be fast. Yeah, sort of, you can't wait for many years to see if there is an effect or there isn't. And, you know, good examples are, of course, these trials that we had for the COVID vaccines and things like that. You need to see the result fast. In Stargardt's, this is impossible. But, and again, what we have done, we have figured out how, depending on genetics, your disease develops. And so we can advise anybody who starts clinical clinical trial, how to do it properly, that you really pick patients that will be informative for that type of clinical trial. So, I pretty much mentioned that. So what is the treatment options? As I said, direct gene therapy, and that is something that I fully advocate for. You just add the normal gene. We did work out gene therapy at Columbia, and it was in clinical trials about 10 years ago until maybe 8 years ago. The issue with gene therapy for Stargardt is that ABCA4 is a very big gene, so it is not suitable for most often used viral vectors called adeno-associated viruses. So these are the vectors. You may have heard, you know, a successful trial and now treatment for a retinitis pigmentosa with RP65 gene. RP65 is a small gene. So you can put it into the AAV, transfer it to the retina, and it works. ABCA4 is too big to put into AAV. So what we did, we used a different virus called lentivirus. And that takes the entire gene, and it worked in mouse models, it worked in non-human primates, and it went into clinical trial. Unfortunately, for some reasons, and I still don't know exactly, but there were some financial and or technical reasons cited. I wasn't part of that trial, that this trial was stopped. I've heard that maybe they will try again, because originally these viruses were produced by a British company called Oxford Biomedica, and I think they still are in business, although I think they were bought out by Sanofi, but you know, I don't know the background because that is already business, and I'm a basic scientist. So it is, I've heard that trials with 2 AAV vectors are coming, so that you put 2 halves of the ABCA4 gene, you deliver both vectors to the same cell, and then they make one functional gene. I heard 3 years ago that these trials will start any minute. I asked the main head of that project to get in touch with me, so we'll be a center. He said, absolutely, I'll be the first one, but I still haven't heard anything, so they have not, for some reason, started those trials. Again, this is a little bit different, but preclinical studies were good. So I hope that that trial will come alive soon. And I also hope that maybe they revitalize the trial that we had done all the preclinical studies for. So then there are other ways of doing, I mean, treatments or therapies. Recently, the last maybe 5 years, the most famous words have been the CRISPR. You know, you do gene surgery, you go in, you replace the mutation with the proper nucleotide, or you do some other tricks. That's a fantastic method, but for Stargardt, it's probably not so great, because the problem is that we have so many mutations that cause Stargardt disease that, you know, doing them one by one is impossible. And as I say, probably for recessive diseases, it's mostly not even that important. But, you know, that's another thing that people are trying. For other eye diseases, there is 1 or 2 gene therapies going on, clinical trials going on with that method. Stem cells, everybody has heard about that, you know, that's great. You just take stem cells, put in the eye, they, you know, become the photoreceptors, the RPEs, you know, you regrow the eye, basically. That is still not happening. It probably will at some point, but the incredible excitement that we had with that about 10 years ago, and actually there are many companies and places in UK that do that. I mean, stem cell treatment, preclinical studies. it looks like that it is more difficult than expected, yeah, to remake photoreceptors. Photoreceptors are neurons, so it is not like epithelium. It's not like growing a new skin that you can do. You can regenerate liver. You can do a lot of interesting things in many organs. But eye is really very complicated, and just to expect that these cells will, when you put them, you know, in the right place, that they will integrate with the current things and start really working is still a bit of a future, you know, things for the future. And then there are other therapies like general survival treatments, what I've already mentioned, and also targeting the functional processes, as I said in Stargardt. These are, there are several planned. Mostly they combat the lipofuscin accumulation, yeah, as the main target for those therapies. Again, they don't target the cause, but they target the symptoms, and some of them are in clinical trials, and maybe some of them will work. Okay, so this probably I already told you. I will not go into this in great detail. One thing is that if you want to treat, let's say, a Stargardt patients, do you need to know they have Stargardts? I mean, obviously you think yes, but then again, it really depends on, you know, which treatment modality are you trying to use. If, let's say, you're trying to enhance cell survival, so you probably, it doesn't really matter. Why I'm pointing this out, that this gene has so many mutations, and even with the current most advanced screening methods, we still lack many mutations in Stargardt patients. And again, I pretty much explained the rest, and I will come about the informativeness of the trial a little bit later, because again, Stargardt is a slow progressing disease, and that is a bit of a problem for clinical trials. So again, you can appreciate these are pictures of the retinas of Stargardt patients, and you can appreciate how different they are. Yes, so, and this is not really different stages, that you'll see on the next slide, but these are, you know, some people have a very severe disease, some people have very mild disease, and the spectrum goes from, yeah, from beginning to the end, and we know now most of the reasons why is that. So this is a simplified scheme here that, you know, the disease can be macular, you know, in the center, you get a hole in the center. Then it transitions, you get those flecks. The flecks are lipofuscin, what I told you before. Then these flecks kind of get together, and you get, you know, really quite major damage to the center of the retina. And then in some cases, as you can see, the entire retina gets pretty much destroyed. There are not many cases like that, but they happen. And I'll explain you also a little bit later. Below here, there are classes of the disease, but I have a better graph where you can see that some of Stargardt patients do not even get into transitional stage. They get into, they stay at the very beginning of the disease, so they never develop into critical and or other diseases. So again, there is a lot of heterogeneity, that means variability, and all the clinical that you saw on the previous slide is caused by genetics. You have over 1,400, now 1,500 new mutations are discovered all the time that cause one or the other form of Stargardts. And we still are missing, if the patient is clinically determined to have likely Stargardts, about 10% of them do not. They have mutations in other genes, but in about 10% still we cannot find the second mutation in the gene. But this is getting much better with the methods of screening we have today. Mostly these are unfortunately still used in laboratories and not in genetic testing facilities. So that's another point. Another big point is that people say, well, it's a very rare disease. Well, it is not. It is the most frequent disease, inherited disease in the retina. And the numbers are as high as 1 in 20 people carry a mutation in ABCA4. So when I hear that, you know, some cases that I had, let's say, a friend, a colleague who actually herself had Stargardt and then was a geneticist herself. And then I said, what do you want to screen? And no, I know I have Stargardt, so I don't care, because whatever, what can you find? You can tell me the mutation. Then she wanted to get married. I said, you know, do you want to screen your future husband? No, I don't care. And then she had a kid and the kid had Stargardt. And she said, well, it's so rare. How on earth that happened? But I said, no, it's not. It's 1 in 20. So it's not that rare at all. So there are currently, although it's not 100% thing, but currently there are ways to plan the family with the current knowledge. So again, and this shows, it's an illustrative picture, that the bottom row is the Stargardt patient eyes. The top row that are absolutely identical are different diseases. Yeah, so that's the point is, clinically, you cannot determine for 100% that Stargardt. You have to do genetic screening. Now, another thing that is, of course, for geneticists, it's quite obvious, but it is quite different in ABCA4 is that not even racial groups or ethnic groups, every nationality, I would say, in Europe has their own mutation per se, that is much higher in that group of people. Yeah, there is a Dutch mutation, there is a German mutation. Again, these days, of course, you know, people admix a lot, so it is not as clear cut. And we work in New York City, that is probably the most diverse city in the world. So our patient population, that we have about 1,000 Stargardt cases in our study, is really, really diverse, but that also has allowed us to figure out specific mutations in specific ethnic and racial groups. Let's say in African descent, people of African descent, the mutations are very different from people of other racial groups. And then you will get into quite complicated analysis after that. Now I still see that we have that funny frame appearing on the slide, but this kind of summarizes, you know, the strength and the categories of mutations we have. That there are about 17% are really bad mutations in ABCA4 that pretty much delete the gene, so you don't have any function. Then there are severe, then there are what we call moderate, then there are mild, and then there are what we call hypomorphs that are really mild. I'll show you a couple of slides how we do that. So I will not go deep into other things, but an important point here is to consider at the bottom there that only 3/4 of the mutations are actually in the coding regions of the gene, meaning that they affect the protein directly. The others are outside of coding regions. What they do is that, for starters, they are much more difficult to find because the gene itself is big. Yeah, it is about 6,500 nucleotides long, but non-coding, and that's the entire locus, that's 140,000 nucleotides long. So screening all that is much more difficult, but about 1/4 of mutations are in regions that do not code for protein. So if you just screen the gene, you immediately miss 25% of the variants. And this is a family that kind of illustrates the point that, you know, that the frequency of ABCA4 mutations is really high. So this is actually a family originating from UK. And you can see that in different members of the family, the phenotypes are very different. And it looks like a dominant disease. Yeah, parents have it, kids have it, so it doesn't look like recessive disease. Now, what is happening is that there are many mutations in this family. So it's not just mother and father with two. So there are very many mutations, and they are listed here. So there are variants in the coding regions, yeah, in blue. Then there are variants in the introns in red. And then there are B copy number variants, large deletions in the gene. So, and this shows you clearly how a different combination, and this is what I call genetic lottery again, will influence the phenotype. If you look at the bottom, so the proband is the middle one that has the 1961 mutation that really and has only a very central localization of the damage. Now, his cousin is exactly the same age to the bottom left and has the retina mostly destroyed. So, but again, if you look at what specific mutations, so they differ for one mutation, and that does such a huge difference in disease onset, progression, and prognosis. So that shows, you know, the complicated issues with ABCA4 and with Stargardt disease. And here are examples of this severe end of the disease where both copies do not work. So you have marked in top red here is a patient that has a very rapid increase in lipofuscin, early onset and rapid progression of the disease. And these are mostly end stages of the disease where some of these even look like here, bottom right, look like retinitis pigmentosa-like. So it is rapidly pan-retinal, so entire retina is involved in these cases. But these cases are not too frequent in Stargardt. Now, the other side are what we call the hypomorphs. Yeah? So that discovery we did a few years ago, and that added the major part of Stargardt genetic clarity, because, and this is interesting, this is, for example, this 1868 variant. This is a very frequent variant. It's in 7% of people of European descent. So if it's such a frequent variant, it's automatically called not disease causing. Yeah, so there is no way, you know, otherwise, you know, half the population will have the disease. And then there are others like that. So what we did, we figured out that it does cause Stargardt if the other mutation on the other chromosome, you know, on the other copy, is bad, deleterious. If one allele is gone, and the other.
Wait, can you just make sure everyone's muted, please?
And the other allele is this very mild or hypomorphic, the disease happens. Now it is a late onset. It usually is diagnosed at the age of 40, 50, even 60, sometimes even later. So this is the other side. And the phenotype is also very different. So what distinguishes this is that you have preserved the central spot, the macula. So the vision is actually really pretty good, 20/20. But, you know, there is still damage around. But if the center is preserved, then the vision is fine. And people don't know until quite late in life that they have Stargardt disease. Now, there are several ways to measure quantitatively the Stargardt disease, and I will not go into clinical details. The issue is that only the area of geographic atrophy is an approved measurement by FDA in clinical trials. And there is a problem with that because a lot of Stargardt patients don't have that. And a lot of Stargardt patients do not progress fast enough to have a measurable difference in clinical trial to see if the treatment worked. Now here is another, I will go quickly through it, another example of Stargardt disease where you get this like a hole in the center of the eye. So it's like we call a bullseye phenotype. And this is caused by that famous 1961 mutation. What is the most frequent mutation in Stargardt disease? In most populations, actually in some populations, it is very, very frequent. Actually, in India, in South Asian population, it's over half of the Stargardt patients have this mutation. And there are some other places. But there are some tricks to this, and I will not go there. But the point here I wanted to make is this mutation, you do not see a huge increase in lipofuscin. So again, if there is a treatment that targets lipofuscin, then patient with this mutation, and as I said, it's a quarter to a third of all Stargardts, do not apply, for that therapy. So these are things you have to know, because again, you can see that, in the middle picture, I mean, there is slight increase. You have the bullseye, there is slight increase in lipofuscin, but it's incomparable to, let's say, this other mutation, that is down the bottom right, where you have a huge increase. So this is another variability in Stargardt disease. And this also, here the graph shows the foveal sparing, meaning the sparing of the center, that you can still see very well, although you have advanced disease. You can see here the picture in panel C, there's a big hole in the retina, but the fovea is spared, and that person has 20/20 vision because of that. So this is another kind of interesting side of all eye diseases. And again, if you have these mild hypomorph mutations like 1868, in panel B, most patients still see quite well into pretty advanced age. 1961 is in the middle, but in most Stargardt mutations, really, you lose the central vision quite fast. Here, the slide shows the progression of the atrophy. And as I said, this is the only quantitative measurement right now in the US accepted for determining success of clinical trials. And you can see that at different stages, and patients with different mutations have very different rates of the atrophy. And that was the question, that if you do a 2-year trial and you use this feature as the feature you detect the effect of a trial or progress, in most Stargardt patients, it will not work. And so that is another thing, that when the clinical trial is designed, you have to get approval for a different quantitative measure. Let's say the same quantitative autofluorescence or some other measures, but this has not yet happened. So that is a bit of a problem, again, in terms of trials. The other thing here is that usually when you start the clinical trial for Stargardt or any other eye disease, phase 1 and even 2 are permitted only on patients that are really too advanced. So because it is kind of the risk-benefit thing, that if something goes wrong, they have not much to lose. On the other hand, you cannot see progress also on those, because you can see just the safety, that you don't get any additional problems, but you do not see progress. So these are these issues with clinical trials that have to be solved in order for them to work better and faster. And again, now I come back to the last few slides to the summarize, that if you see, we had these grades, stages of Stargardt disease, but you can see that if you have certain class of mutations like this 1868, you will be in this first stage, maybe in second. You will not, develop advanced disease. 1961, the same, you can go a little further, but then the disease is a little earlier. But then the kind of median Stargardt, what I call classical, is the third, what is the other ABC4 alleles, but that also takes long time to develop. What is good news for Stargardt patients, because I was hoping already at least 10 years ago when we did the gene therapy preclinical studies that it will advance in the clinic, no, let's say in 5 years. But unfortunately, due to many reasons, this has not happened, and this has frustrated me. But the point still is that in most Stargardt patients, even if they have relatively advanced disease, there is hope, yeah, that a treatment that comes finally and is approved will have an effect and help them. Of course, in the worst situation are those with very severe disease where there is null ABCA4 activity, meaning there is no functional ABCA4, its onset is early, disease develops rapidly. In those cases, if the treatment cannot be applied, you know, in the mid stages of the disease development, I hope that stem cell treatment still advance to the point that you can replace photoreceptors and RPE and the cells that have died. And so we have made this kind of a matrices where we can predict how the disease develops based on the genetic knowledge. So we know what the mutation is, and we can predict the progress, the severity, and also separate specific subgroups to deliver for specific clinical trials. And this is what is very important, that you really, when you enter some, I know that patients are of course desperate and they want, you know, to try anything that becomes available in terms of trials, but I always advise those people who direct the trials to be very honest and say that, okay, this is what you could expect. Yeah, so do not really get hopes too high. And the other thing is what's still happening is that clinical trials that are ongoing take in patients that theoretically will not see any effect, because that's impossible based on scientific knowledge and clinical knowledge and genetic knowledge, what we have. So, last slide. Do you need to know that you have Stargardt? Yeah, you do. So if you are enrolling in a clinical trial, or if you are thinking, looking for a treatment once they are approved, of course, you have to know that you really have Stargardt disease, meaning you have to have 2 mutations that are on 2 different chromosomes. Again, as I said, I'm the big proponent, again, theoretically, as a geneticist, as a scientist, of gene therapy. And I would really like to see these to work. And I talked already about that. Now, the other treatments, yeah, whether it is the Allikmets treatment, whether it is, you know, similar treatments, they will work on some patients, probably. But there are some other issues with those, I can tell you in the question and answer. But you could see, let's say, my examples of patients with the disease with a 1961 mutation, they do not accumulate lipofuscin. So I don't think that those patients should get this treatment. On the other hand, if there is nothing else, you know, that will probably do no harm. And then again, I already discussed the issues with trials, and we can do a lot right now. We know a lot, and I am surprised that, you know, when I cloned the gene 25 years ago, I thought, okay, it's another gene, and for a while I didn't really work much on that. I mostly worked on age-related macular degeneration. But then, yeah, when genetically looking deeper into it, and since at Columbia I'm at the ophthalmology department, so we have clinic integrated with basic science that I direct. So I understood that the Stargardt disease is a much more complicated, both clinically and genetically, entity. And so the last 5 years, we have made huge progress in kind of figuring out the basics. Yeah, before, when you ask me, you know, patients ask, okay, what do you predict, or what would my progress or be? I said, I have no clue. And now I can do that pretty well. And I am not a clinician. So but I can do that kind of a theoretical predictions. It's much more complicated than that, because now we're looking at modifiers of the disease, and we have found many, and so I will not rack your brains with that information. But the positive information is that, yes, we know a lot about what causes the disease and how the disease starts and develops and progresses. There are many clinical trials out there. Unfortunately, none of them, as far as I know, currently does gene therapy, but I hope that will happen sooner than later. And we are working. We have made very good progress in this field, although I am frustrated that treatment options and clinical trials do not develop as fast as one would think. And so maybe one role of, the patient groups like yours would be kind of put this pressure a little bit on researchers, who develop clinical trials, to kind of get them to move faster, so to speak, because the rules and regulations are bad. But little nudging always helps, and I've seen that in my life. And and sometimes you don't need little nudging, you need a good kick. But so that is kind of the summary that I wanted to present. If something was not clear, you have the presentation. I can answer questions. I can answer questions now. I can answer them, let's say, later, if Balna collects questions and sends to me. So in due course, I can do that. So thank you very much for your attention.
Thank you so much, Professor Rando. That was absolutely fascinating and really, really informative. And thanks for your brilliant work that you've been doing. It makes us all feel really hopeful. Did you want to go to questions now? Or did you, how did you feel? Or did you want
Yeah, we can start now, I guess. Yeah.
All right. We've got some in the chat. Rhea, did you want to start them off? Or did you actually, Rando, do you mind?
Let me try.
If you sort of come out with your screen share, so then we've got everybody.
Yeah,
all right.
I'm trying to figure that out, but there is a little bit, let me see. No, stop share. Okay. Did that work?
Yeah, it did.
Very good. So I'm not sharing anything, am I?
No. Fine.
Okay.
Shall I start off with some of the questions? There's many of them you've actually answered, but there's a whole range of questions around what exactly are the benefits of genetic testing, and you need to understand exactly which mutation you've got and which mistake you've got on your genes, and are there more mutations likely to be found, particularly in countries where they're not doing genetic testing and people might have Stargardt, and does the actual fault on your gene dictate the progression of the disease or the severity of the disease?
Yeah, I think I pretty much answered all these questions during my talk. Yes, you need to know mutations, because again, as I showed, and I think there are a couple of answers that I see, a couple of questions I see later here, is that people are asking, let's say, the other genes that are mutated in diseases called Stargardt-like, and I am always very strict that there is only one Stargardt disease. Unfortunately, Stargardt-like is a misnomer, and this is in the literature, because they are not really much Stargardt-like, but they were called like that, and that's why they are still considered. So diseases with ELOVL4, PROM1, they're totally different diseases. I mean, there are phenotypes that overlap, and this is what I mentioned in my talk. There are phenocopies, like we call it. Yeah, when you go to the doctor looks at your image and says, no, you have Stargardt. And then we screen that person, and we screen them very thoroughly, yeah? Better than most diagnostic testing centers. And we don't see any mutations in ABCA4, but we find mutations in some other genes. And a very famous phenocopy is the gene for pattern dystrophy, which is RDS peripherin, yeah, or PRPH2. These two diseases are very overlapping. Clinically, they are, and that was an example of one of my slides, they are indistinguishable clinically. So that's why you need to know your specific mutations. And screening has become much better, even the diagnostic testing centers. But there is still this gap that they don't do what we do in the lab, that we screen the entire locus. Yeah, we screen coding, we screen non-coding, we look at regulatory regions. So there are many, many things we do that is usually not done at a diagnostic center. So, as I said, the last few years after discovery of those hypomorphic mutations, yeah, that are very frequent, and after discovery of many others, our percent of solved cases genetically has risen well over 90. It was 75 just a few years ago. So we can detect most mutations, but again, it depends on the screening methods, on the screening center, etc. The other thing is that interpreting the data, what you get, is very important. A lot of places still say, well, this is not a mutation. Patient says, well, Dr. Allikmets says it is. They said, oh, he's a geneticist, he's not a clinician, he doesn't know. So I get those comments all the time. So it is very important to do a thorough screening, but it is also very important to get a good consultation. That, okay, this is what was found, okay, what is the deal? But it's not 100% yet. So I am clearly knowledgeable of that, and the point that in some racial ethnic groups where screening has been less thorough, yes, there are new mutations. New mutations are coming up a lot. There was recently several papers, say, from China, there are papers from India, other places like that. In Africa, there is not much done at all. Yes, and new mutations, yes. Sometimes they say, well, we found 10 mutations, 5 of them are new. Yeah, so this is increasing. However, our knowledge if this is a mutation or not is also increasing. So we can usually determine that.
Thank you for that. And the next group of questions are very much around the treatment. How exactly does gene therapy work? For example, does it replace the faulty gene? And I think you've addressed it in your talk, the difference between gene therapy, which does it actually stop the disease, or does it actually help improve the disease? And the difference between gene therapy and stem cell therapy.
Yes, they are very different things. So gene therapy for Stargardts is not replacement, it is addition. So sometimes it's called gene augmentation. You don't need to replace it, because if you have a faulty gene, it doesn't work. You know, the protein doesn't work, or it works a little bit. So you just add the functional gene that makes the functional protein. So that is the gene therapy, direct gene therapy. And that is, the question is again, you know, what would be the end benefit is a good question. We need to know that. It certainly, we hope, stops the disease. Will it improve? It's difficult to say. It might. Again, it will not completely cure it, I understand. I mean, again, it depends on when the treatment is applied. Yeah, I mean, if it's advanced disease, then it has, of course, less possibility to have a great effect. But this is, yeah, gene therapy is really the, you know, available method right now to apply for Stargardt disease. It is not that complicated. Now, the other is what I said, stem cells, etc. These are much more complicated things. There are many other issues that are, because first I said they don't work yet as they are hoped they will. Then there are issues, and unfortunately, another thing what I want to caution, especially patients, is there are all those stories around that, well, there is a great Stargardt treatment, they take your stem cells and from you and put it in the eye and you'll, improve drastically. And this is total baloney. So this is absolutely wrong, because you will not cure anything with your own stem cells, because these cells have the same mutations that you have, because these mutations you have. So all these stories that, yes, there is a center in, Russia or what, in the US we have the same. You have those clinics that, promise great help for Stargardts, but they have, it is a snake oil. However, theoretically, yes, you can do the stem cell treatment if you take your own cells and then modify them, correct the gene or add the gene in the dish, and then you put them back. So that's one way. The other way is to use the cells from healthy people. But what I'm saying is the main problem with that currently is still that they will not grow in and make the new retina yet. There have been some progress. They may work at some point. Now people are trying, instead of just spritzing cells, the cell suspension, they're trying to make sandwiches of cells, photoreceptors, RP, and then implant that, and that works better. I know in age-related macular degeneration, they are doing that. So all these methods are advancing, but yes, even to me, but definitely to patients, little too slowly.
Thank you very much for that. There's a specific question here about, do you know the institution conducting the gene therapy trial using Oxford Biomedica Sanofi?
As I said, this trial was stopped.
It was stopped. Okay.
And I was told that there were, some technical and monetary reasons. I was also told that it may start again, but that's all I know, because that is the trial that, not the trial, but that's the method we did at Columbia. So I worked with Oxford Biomedica, he provided the viruses, we did the work. So it could be that, because this is the delivery by lentivirus. Lentivirus takes the entire ABCA4 gene. And I've heard that actually the two AAV method that I also mentioned that should have started already the trial, I believe still in lentivirus more. And I also have heard that from several people in Europe that they are working still on lentivirus-based treatment for Stargardt. So if the Oxford Biomedica will reopen, or I don't know what their situation currently is, or other will work on lenti or even the two vector things, they should advance. Yeah, but that's my current knowledge.
Okay, thanks for that. And then a couple of questions around clinical diagnosis of cone rod and 2 ACA4 mutations and ELOL4 or PROM1. Should they be considered Stargardts?
No, this is what I said before, that these are different diseases.
Okay.
ELOL4 and PROM1, they are called Stargardt-like, but they are totally different diseases caused by totally different mechanism. And so the treatments for ABCA4 disease, that I now call Stargardt, because just a part of ABCA4 disease is Stargardt. So there is cone rod dystrophy, there is, pattern dystrophy, there are many, many clinical terms. So ABC4 disease is specific to this ABC transporter. The fault is known. It is the transport of vitamin A is not working. As I said, many treatments are designed to kind of alleviate that. There is a question I saw that, is it important to avoid vitamin A rich foods? Yes, that's what I say. Again, I don't think it will have a major effect per se, but I think that, you know, that notion by grandma that eat your carrots, you'll see better is not correct in Stargardt. It is correct in some other eye diseases, because here the problem is that you have too much vitamin A. Yeah? So ABC4 is not working, it's not kicking it out of the system. So that is really the problem of most Stargardt cases. So yes, avoiding vitamin A is good. When I started this field, I heard that a lot of doctors prescribed actually vitamin A supplements, and I said that's totally detrimental. You know, you don't do that. So, but again, it will not probably have a major effect. I mean, the two things that one can do and what are obvious is avoid vitamin A. You have plenty of vitamin A, so you will not be vitamin A deficient. I mean, it's practically impossible, even if you completely don't eat anything, but because you get enough. And the other is avoid direct sunlight, daylight. Yeah, so shield your eyes from light. You know, UV blocking sunglasses. Really, because the issue is that, and this works with many, the treatment is that you try to slow the visual cycle, that you will not get a lot of these side effects that lipofuscin accumulation. And so that is kind of theoretically how it works. And these two things, because if you close your eyes, your visual cycle will not work. Yeah, I mean, as much definitely. And so there were even anecdotal things. People said that they were trying treatments that, you know, you kind of block light to one eye. Yeah, so you, for prolonged, so you use one eye and then, you know, this eye will not get worse, and then you'll, you know, do that. But these things are too drastic. So that is kind of a general suggestion, yes, that avoid the light as much as possible and to the eyes, yeah, blue blocking, UV blocking sunglasses, and vitamin A. So that's the answer to that.
Thank you. There's a range of interesting clinical presentations here, which I'll just go through very briefly. Somebody's asking, is it a normal thing to happen if they have two children, one whose onset of Stargardt's is very early, very young, and the other whose onset is a lot later? And then somebody who's diagnosed in their 20s preserved their sight until in their 40s when their sight started to deteriorate. And then this one I'm going to have to read. It's very interesting. I have 3 questions. My cousins, brother and sister, are married to non-relatives. One of the spouses is from a different country. Each of my cousins has one daughter who was diagnosed with Stargardt disease around 15 and 18 years of age. I know that the gene has come from both the mother and the father. Is that possible that they both, her cousins or his cousins, had the gene, and also their spouses had it too? Very interesting, that one. Are they good candidates for gene therapy at this age, etc.? So clinical presentations.
Yeah, again, I have to say right away that I'm not a medical doctor. Yeah, I'm a PhD and a basic scientist. But what I know is, let me start from the probably last part of the question. I showed this family from UK that is totally unrelated people. Yeah? I mean, some has Irish ancestry, some Scottish, some English. So, but they are not related at all. And they have many mutations, yeah, in this one family. And that's how you get many different disease presentations. Because again, I showed these 2 cousins, yeah, one had the 1961 mutation, had a little hole in the center. They were both at the same age. My guess is like 30s at that time. The other cousin, the same age exactly, had a different mutation. One mutation they had the same, but the second was different. And that the other mutation made a huge difference in clinical disease presentation. So that is the answer, that this is, I mean, any disease is really a genetic lottery. Yeah? So you don't know until you see it. If you have it in the family, testing will help greatly. Because that I always say that if, let's say, no, I don't give that advice because by law I am not allowed to patients, like genetic counseling. Yeah, there is a patient of Stargardt disease saying, I want to get married, I want to have kids, what should I do? No, I said that, you know, screen the spouse. But again, I warn, no screening is 100% still, you know, so it's still, there is a chance you miss a mutation. And so that is another thing you have to do. But you can do quite a bit. And then you can do, you know, in vitro and, you know, select the proper combination of chromosomes. Because if you are already a Stargardt patient, so your kid will have, you know, either of your mutations by definition. So the kid will be heterozygous right away. So again, the mutations in ABCE4 are very frequent in population. I said 1 in 20. Not all combinations cause the disease. So that's another big point, because people said, well, if it's that frequent, you know, we should have like 1 in 100 have Stargardt. Why it's in 1 in 10,000? I mean, it's more than 1 in 10,000, but so the mutation rate in the population is frequent. In terms of, we know also how different combinations cause different diseases, different onset and different progression. The question about the same family, let's say 2 siblings having different course of the disease, that I have not seen. There are many papers, and actually I think I even published one that we said that there is discordant development of the disease in a family, but usually it is still explained with you know, a little bit different time course and things like that if you adjust for age. Another thing is that, you know, usually the first sibling is diagnosed much later, yeah, well into when the disease has developed, and then people already know that, okay, let's take a look at younger siblings, and they get, you know, especially clinically evaluated when they are asymptomatic, but the doctor may see already something. So I don't think that there are many cases at all of siblings having a different disease. Yes, there is some difference. I mean, even siblings, even twins are not totally identical. Yeah, the maternal twins, they are still is different. We all have, you know, several thousand variants in our genomes that makes us different even from a sibling, even if they are from the same parents. So this is kind of the answer, that a thorough genetic screening would explain most of it. And so then you'll take it from there.
So I think the message is about thorough genetic testing. I'm just checking, are you all right? There's a couple more questions and a couple of people who got their hands up. Are you okay to go on for a bit, or do you need a break?
Well, let's do a 2-minute break. And
yeah, no, that'd be great. The other thing is, if we can't answer all the questions, are you all right if I send them to you and we can post them, like maybe on the website or something, or we can post it to
oh, you can, yeah, send them by email, or again, answering questions in writing takes time, and that's probably
say that again, sorry.
Problem. But I can get them, and you know, we could do a, you know, separate Q&A session, you know.
Maybe we can do that, because I think there's so much. I mean, it'd be great to have you back.
Yes, because there is, you know, I understand there are lots of questions, and well, let's do a 2-minute break. I'll answer some more, and then we'll see how it goes.
And then what we'll do, because I did say there was going to be breakout rooms. It's just, you know, so I'm just going to make sure that we have a little time for people to break out as well. But yeah, if we have a little break, and then we can, yeah, just
Okay, 2-minute break.
Okay, brilliant. Thank you.
Thank you very much. So we'll come back in 5 minutes, because 2 minutes isn't enough time. So we'll be back in 5 minutes, everybody. is saying that she is a co-founder of Revision Therapeutics in NYC, and they're developing a drug for Stargardt that removes lipofuscin. Gene therapy will not remove the lipofuscin. Is that correct?
Gene therapy will stop lipofuscin from forming. I have heard about these options where people are trying to remove lipofuscin. Again, in my presentation, I mentioned two things. First is many Stargardt patients do not accumulate lipofuscin much at all, more than normal, healthy people. So in that case, it is not applicable to them. Scientifically, there is also a big discussion. You know, lipofuscin was determined as earlier as the main cause of Stargardt disease. It has been disputed quite a bit now if it is so or not. I still think it is in many cases. And removing lipofuscin theoretically would help in some cases. But again, you're talking about targeting the downstream effect. You're not talking about targeting the cause. Gene therapy targets the cause, that you put the normal functional gene in. Because removing lipofuscin is similar to somewhat to the other trials that are trying to stop lipofuscin formation, such as, you know, Allikmets trials and the others. They are trying that by different means. Some are trying to slow down the visual cycle. That's what I said. This will work similarly to, let's say, not eating vitamin A or not exposing your eyes. And there are different ways of doing it. Again, one word of caution is that oftentimes these type of treatments are very effective in model organisms, in mice, for example, but in humans, the situation is different. That goes for any drug, it goes for any disease. I mean, we have cured cancer in mice in many, many times. It is oftentimes does not work. The same is due to Stargardt's often. Let's say the Allikmets thing, that was also worked out actually in our department, not by me, but by other people. So they are modifying vitamin A, so it will not form lipofuscin or A to E, the main component. So what that means is that the patients are given a specific vitamin A supplement that is modified. So you will replace your natural vitamin A with a modified compound. Now in mice, that's very easy to do. In humans, that's a little bit different. You can take mice and put them on this diet and they are in controlled environment. They don't eat anything else. In humans, it's a little different. So again, the other point is that you start treating, model organisms, from birth or from early stages. That is not the case with Stargardt disease. So again, every therapeutic option targets some stage of the disease. They all have a chance to work, but why I'm saying that gene therapy is the best, because you get the functional protein in it. Now, again, as I said before, it depends at what stage of the disease you get it. And science and treatment is always empirical. You don't know exactly what effect you have until you do the study. And then you'll see if it will stop the disease, if it will slow the disease, if it will actually work in the way that it will improve the condition. And again, it depends on so many factors, starting from the time when you start the treatment, and many others, that it is not always easy to predict. I'm just talking about theoretical possibilities. So that is my answer to the questions about treatment.
Thank you so much. I think we may have answered Daniel's question because he's put his hand down. Can I just check, Daniel? You've put your hand down, so I'm assuming we've answered your question. So if that's the case, I think there are a range of other questions that if we could post them to you and get answers that way, that would be really helpful.
Yeah, I think that's fine. I can quickly answer a couple of other things here that I see that.
Daniel's got his hand up again, so I don't know. I'll unmute you. Are you on? You're on mute, Daniel. If you want to answer.
Yes. May I speak here?
Yeah.
Thank you. I'm a clinical geneticist from Panama, and the talk was very nice. I just wanted to ask just a couple of things about the treatment. What about using gene therapy to target life function to directly direct it to the lysosomes? The question is, what about using other vectors like can carry bigger quantity of genetic material like JAKs or BACs to as vectors? Thank you.
I did not hear the first question very well. In terms of other vectors like JAKs or BACs, these are not very easy to use in humans. Again, there are many things you can do in model organisms, because that's why viral vectors are, because these have been worked out for a long time for different diseases. And so they are most efficient for the eyes. Now, there are other vectors that are tried. There are nanoparticles, there are other means to deliver, and these vectors are being examined, and if they are efficient, because there are many issues with treating a human eye. Yeah, one is that how efficiently will this vector deliver the normal gene, to the eye. The other thing is that what about possible side effects? Because with gene therapy, some of you may know there have been, history of real mistakes and mishaps that slowed the gene therapy progress for quite a while. So any method is in process of development. And so, as I said, currently for gene therapy, I don't think that the JAKs or BACs will work here for many technical reasons. The first part of the question, I'm not sure I heard.
I can repeat it. It is that maybe a lipofunction could be targeted to the lysosomes as a lysosome metabolite and be decomposed there. Will it probably by using gene therapy for that?
You don't use gene therapy for that. There are other ways of, because now that question I think I answered before about, trying to get rid of lipofuscin. Yes, there are ways of doing that. Again, these are methods are being developed, and we'll see. Because yes, preventing lipofuscin from forming and also eliminating lipofuscin is two ways people are trying to also treat Stargardt disease. So, but that's not exactly the gene therapy based approaches.
Thank you so much.
There was another series of questions about genetics. I understand that, the inheritance and how children will be. Now, that takes a little time to explain. However, again, the math here is quite simple, theoretically. If both parents are carriers, they of course don't know that they are. So then it is, the kids have 25% of chance to be affected. Again, this is theoretically. I've seen families where, you know, there are 4 kids and all of them are affected, because that's, again, genetic lottery, how the chromosomes come together. I've seen families the opposite, that there are 10 kids and only 1 has Stargardt. So, because in every time you start the kid, you have 25% of chance of the kid being affected, just by chance genetically, if both parents are carriers. Now, if one of them is a Stargardt case, is affected already, then the odds are, you know, 50-50. Yeah? So because from the affected parent, the kid gets one of the 2 mutations, and then it depends. If the other parent is not a carrier, then there is nothing wrong. And in most cases it is so, but if they are, then it's 50-50 chance that the kid will be affected.
Thank you so much. I think this has been absolutely amazing, and I can testify to that by the number of questions. This is the most questions we've ever had in one of these sessions, so it has been wonderful. Thank you so much, and we will be sending you some more. I can't tell you how grateful we are for you getting up this early for a start, but to
Well,
come.
it's not that early, you know. So.
Oh, good.
I mean, it was early for Saturday, because, you know, I got up like
I have one question.
regularly for a job, so that is that's not that early. Now, as I'm saying, I'm always pleased to help if any way I can. I used to do a lot of these presentations to the US patient groups some time ago. It has been a while now, but again, I have to say that especially the last 5 years, our knowledge has really developed big time. And that's why I hope that treatments will be coming. I see, there was another question that Moorfield says it takes 10 years. Nobody knows. Yeah, I thought 10 years ago that we are 10 years away. So it depends. And again, since clinical trial and drug approval, that takes a long time, unfortunately. As I said, maybe, you know, with this COVID thing and others, maybe now even other diseases get, you know, a little quicker regulatory paths, yeah, forward. And so that would help, because, yeah, I see absolutely no reason why there is not already a treatment or 2 out there for Stargardt's, because, but any kind of guesses is as good as anybody's guesses. Yeah, we'll see how these go, but clinical trials take time, and they take years and years. And I mentioned about Stargardt, the progression is slow in general in most people, so that's another reason why it takes many years to see if there is an effect or not. And so there are many associated questions. And then there was another question about what about Stargardt's with 3 mutations? That's the same as with 2, because then the 2 mutations are on 1 chromosome, 1 is on another. And 2 mutation chromosomes are at about 10%. So it is not that rare also. But then again, it is still 1 + 1. You know, in that case, 2 + 1, but it is genetically the same as 1 + 1.
So I thank you so much for that. I think the other thing that you reminded us of is the power of patients and patient organizations like this. And Stargardt's Connected has grown exponentially. We're now an international charity with these Zoom sessions, with hundreds of people joining us. So we will use that power to support all the amazing researchers who are looking into Stargardt's, and we will be sending you some more questions if that's all right. We're now going to break up into one of the powers of these Zoom sessions is people getting together, particularly newly diagnosed people. So we're now going to move into breakout sessions.
I have one question.
Can I just say what we could do is we'll definitely have you back again if you don't mind, because I think if that's okay with you. It'd be great to have you back, maybe for like a question and answer session, but it's been so fascinating. So thank you so much. And we will put up the recording on the website as well. And any
I mean, if you make a list of questions and, you know, we have a Q&A, so that's fine. I understand that the same questions will come up again, and, you know, so it is.
Okay.
I see here there is a comment about genetic testing through Foundation Fighting Blindness, my retina tracker. My point is that, yes, there are many places that provide genetic testing right now. However, they are not all on the same level of thoroughness. So that's one thing. And the other thing is that explaining the results is the most important part. And that is still not up there yet, yeah, because people go by standard that, you know, this was published as mutation. I say, well, it was 5 years ago. Now it's not a mutation, or vice versa. So that is a big part of the genetic testing, that you have to have it done very thoroughly, meaning the entire locus. And the other thing is that you have to have a very knowledgeable person to explain the results. That these are two things that have to develop still further. Okay, so it was.
All right, thank you very much, Professor Rando, and we really appreciate it. So thank you.
Thank you. You have been amazing. Thank you so much.
Thank you.